Good afternoon, everyone. And like the title poses, the question is whether mass spectrometry has the ability to diagnose TB drug resistance. Now, to answer this question, ladies and gentlemen, we need to first look at what is routinely used in the tuberculosis laboratories to diagnose TB. And yes, we are looking at acid-fast stains here of submitted TB patient or potentially TB positive patients. And routinely in the tuberculosis laboratory, an acid-fast stain would be performed. We would see either numerous or moderate acid-fast bacilli, and we can even see scantily stained acid-fast bacilli. And based on 2 of 3 positively submitted specimens, the clinician or physician would diagnose the patient as having TB. But afterwards, you would still need to have a culture done on the submitted specimen, as well as an identification and a drug susceptibility testing. With molecular and PCR tests now widely available, they are of course expensive, but they offer the rapidity and accuracy of determining this or determining this answer, the drug sensitivity answer, at least in a shorter period. Our particular research of course focuses on the phenotype of the organism. And here I show you a cross-sectional diagram of one of the bacilli. What we are focusing on in our research is the thick cell wall that exists in many of these mycobacterial species. If one takes a closer look at the cell wall, you will notice several fatty acids, long-chain fatty acids, as well as glycolipids and peptidoglycans that exist in the thick cell wall. And these have a different profile between the different mycobacterial species and potentially the different strains of mycobacteria that exist. So our current research is going into the finer details of the chemistry of these lipids that exist in the cell wall of mycobacteria, particularly Mycobacterium tuberculosis. And the most prominent lipid that is present in the organism are the mycolic acids. And here we show the mycolic acids that can be divided into 3 main classes, the alpha, keto, and methoxy class. And these 3 classes have the similarity of having at the carboxy end, or the carboxy motif, which is a part of the branch of any of these long-chain fatty acids that can be carbon 60 to carbon 90 in length. At the proximal end to the carboxy motif, there is always the presence of, my apologies. At the proximal end to the carboxy motif, there is always the presence of a cyclopropane ring. At the distal end of the chain, there is either a second cyclopropane ring which distinguishes the alpha mycolic acids from the others, but then the keto and the methoxy mycolic acids have either a ketone functional group or a methoxy functional group. Now, the keto and the methoxy class of mycolic acids can exist in either the cis or trans configuration in nature, and this is with relation to the positioning of the cyclopropane ring in relation to the functional group at the distal end. The earliest type of chromatography was done in the 60s, 70s, and 80s by David E. Minikin and Watanabe. Well-publicized experiments showing thin layer chromatography and essentially normal phase chromatography, separating these mycolic acids from each other, so a single dimension or even a 2-dimensional TLC plate can be performed. Then, as far as the 90s and the early 2000s are concerned, a well-implemented test in the laboratory to speciate mycobacteria is an HPLC fluorescence assay, and Butler and Goutet, in collaboration with the CDC, optimized and validated, in fact standardized an assay that was well implemented globally to identify different mycobacterial species. This was performed routinely in the microbiology laboratories. Very easy for a microbiologist to look at the profile and compare the different profiles of the species, but of course a chromatographer or analytical chemist will come along and say this chromatography does not have sufficient resolution to identify the eluting compounds. Nonetheless, this was performed for almost a decade before the molecular and PCR tests came along, and because of their rapidity and accuracy as far as confirming the genotype is concerned, not the phenotype, but as far as confirming the genotype is concerned, took over in the laboratories. So, the aim of our particular study was to develop a selective and sensitive accurate mass liquid chromatography mass spectrometry method to identify and quantify each of these classes of alpha, keto, or methoxy mycolic acids, which can then be used to differentiate between different mycobacterial species and possibly strains. Some of the objectives, and I won't run through all of them, but picking on some important ones, was to determine the ratio between the three main classes, the alpha, keto, and methoxy mycolic acids, and compare them between what is prepared in the laboratory, and this was done in collaboration with the Department of Biochemistry at University of Pretoria, and they prepared and isolated mycolic acids for particular downstream application that they have. We then also ordered commercially available mycolic acids, which is also a different species from the MTB H37RV strain that was used in biochemistry. So the commercially available one is the Mycobacterium bovis strain. Another objective was to compare a triple quadruple mass spectrometer to a time-of-flight mass spectrometer. So essentially there we're comparing nominal mass acquired data to accurate mass acquired data. So just to touch on how it's performed in the laboratory when a patient's sample is submitted, yes, we perform the light microscopy, but what also needs to be done is a TB culture, liquid culture these days, after which the identification, as I've said, and drug susceptibility testing. So this is how it looks in the laboratory, the TB culture liquid system. Some of the personal protective equipment that one needs to use, especially when working with multi-drug resistant and extremely drug resistant TB. We're talking of here gloves, laboratory coat, decontamination suit, followed by a second glove, headgear, and face gear. This is just to give you an idea of how these cultures look after subculturing them on solid media, and we actually worked on solid media, collected bacterial cultures this way for our extraction and analysis on the mass spectrometer. The most recent part of this study was a collection of 4 different categories of mycobacterial resistant strains, a wild type compared to single resistant, either rifampicin or isoniazid resistant, and multi-drug resistant TB. So in brief, these were collected in 2 different types of solvents. We collected in a potassium hydroxide solution to saponify the mycobacteria. We also collected in distilled water, and both of these collections were autoclaved at 130 degrees Celsius for an hour. And then after cooling down, we did a chloroform methanol extraction to get at least 2 layers here. The bottom layer contains what we would want to look at, the mycolic acids or dissolved lipids, and this is the chloroform layer. The top layer, consisting of the methanol chloroform, will contain most of the polar metabolites. There is also a middle layer which is very important, which contains a lot of the protein debris. So the chloroform extract was transferred to a separate tube, dried under nitrogen, reconstituted for examination or analysis on the mass spectrometer. Now, the study we performed with the biochemistry department includes a lot of mass spectrometry, liquid chromatography and mass spectrometry on the triple quadruple mass spectrometer. What you see here is a mass spectrum as used with their study to differentiate between the alpha, keto, and methoxy mycolic acids. But in this mass spectrum, one can see that the keto and methoxy mycolic acids are overlapping, which is why liquid chromatography needs to be put at the front end of the mass spectrometer to resolve or separate these compounds. Why we used just the parent ions or primary ions in the method instead of the product ions, one can see here that a product ion scan of any one of those fragments reveals product ions that are unfortunately not unique to any one of the parent ions, and this can be seen by performing a precursor ion scan, where a precursor ion scan shows that each and every one of the parent ions come up in the scan. Moving over to the liquid chromatography that had to be used to be able to separate each of these ions and then quantify or integrate the areas under them for the study. In brief, C18 column was used, 15 centimeter, 4.8 millimeter diameter with 3 micron size particles. A gentle gradient was used, and mobile phases here were methanol to methanol chloroform with ammonium acetate and formic acid as additives. So the study with biochemistry ended up in, or resulted in a relative ratio difference that we could obtain using either the dominant peak from each class in the chromatography and integrating that, comparing the Mycobacterium tuberculosis strain to the Mycobacterium bovis commercially available strain. And we do see a difference between the methoxy and the keto classes of these strains. So everything up to there is a paper in preparation for submission as early as the end of this year, I would hope. I'm going to run through the next slides very quickly because I'm out of time. But the current analysis we're performing on the time-of-flight mass spec, also at University of Pretoria Chemistry. And here you see one of the multi-drug resistant strains, the entire range shown here from 50 to 3,000 mass to charge ratio of the parent ions there of mainly mycolic acids, or what we think are mycolic acids. And here we see preliminary runs showing the difference between INH and or isoniazid and rifampicin sensitive strains compared to the mono-resistant and multi-drug resistant strains. So the data is yet to be crunched to determine ratio differences between the three classes identified there. This is to show you the difference that we find in the potassium hydroxide extracts compared to chloroform methanol. What you're seeing here is actually quite novel, a Van Crevelen plot of a hydrogen-carbon to oxygen-carbon ratio, which has not been reported as yet. This is not on experimental data, on theoretical data. If we zoom into that little area over there, that has not been shown before, and you're lucky enough to see this right now. On theoretical data, remember theoretical data, not experimental data, showing that there is possible separation between the three classes of mycolic acids if one looks at it in this way, and taking into account, if we calculate the mass defect values as well as retention time as well as signal intensity, one can potentially find a difference between even different strains of mycobacteria. So I won't run through any of these conclusions because I've discussed it through the slides and I'm running really out of time now. Just like to give some acknowledgements to Professor Duncan Krumati, my supervisor at Department of Pharmacology, Professor Jan Verskoor at the Department of Biochemistry. Both of these professors are present in the audience, so ask them all the questions, please. And Dr. Yolandi Lemmer, senior researcher working with me at the Polymers and Composites Division at MSM. I'd like to thank my family, Dr. Hulda Swai, who recently retired from the CSIR. Many of you know her. I thank her very much for welcoming me into the nanomedicine group at PNC. And yes, I'm making the best of that opportunity. My lovely wife and my son. Thank you very much.
Thank you. Very interesting.